All right, class, this will be our last lecture for lecture number 9 and also our last lecture for unit number 3. So before we get started on our last mechanism of recombination, I want to talk a little bit about some terms that I had previously discussed in the beginning of lecture number 1, or the first part of lecture number 9. So remember that we can have media that has that we can either consider media complete or minimal. And minimal media means that it doesn't contain the nutrients needed by this particular experimental organism. So if we have an organism, that needs, say, tryptophan in order to grow, minimal media would not contain it, whereas complete medium would. So don't get this confused. Like, for example, glucose salts is most certainly a minimal media because it's going to lack pretty much any nutrients. But it's not necessarily, it doesn't necessarily have to be glucose salt. It has to just simply lack that one key nutrient that particular bacteria needs. In other words, there's lots and lots and lots of different kinds of minimal media depending on the particular kind of experiment that we're setting up. On the other hand, let's take a look at the bacteria. Remember that bacteria that are able to synthesize all of their own amino acids and all of their own cellular machinery are considered prototrophs, and they're able to grow on minimal media. These are also called wild-type bacteria. Wild-type E. coli is a perfect example of this. E. coli can make everything that it needs in order to grow, even if it's growing on glucose salts broth, which of course is a type of minimal media. On the other hand, if we mutate that bacteria such that it can't make its own, say, amino acids, that bacteria is now considered an auxotroph. In other words, it cannot grow on that minimal media, but it needs some type of supplementation. So if we were to take an E. coli, a wild-type E. coli that's a prototroph, mutate its genes such that it cannot make its own tryptophan, it would now be considered an auxotroph and would have to be grown on complete media that contains the amino acid tryptophan. So we're looking at, in this particular case, we're looking at the ability to generate, or to what would it be, and the ability to anabolize the amino acid tryptophan. So the genes that are responsible for that anabolism would be considered our genetic marker. All right, so now let's talk about a really interesting experiment that Lederberg and Teddem's group did in 1946. They took, there's a lot of information in this slide, so let's just take it kind of piece by piece. Let's look at the left-hand side of this slide first. So they had some bacteria, it's E. coli. They had some E. coli bacteria that was a mutant strain of E. coli that was unable to make its own tryptophan. However, it was able to make arginine. There's another type of amino acid called arginine. And so when they took this bacteria and they plated it onto media that does not contain tryptophan, as expected, this bacteria was unable to grow. On the other side of this slide, they took another mutant strain of E. coli that could make tryptophan, but it couldn't make arginine, and they plated this onto media that does not contain arginine. So as expected, we get no growth on either one of these media because the bacteria can't make a particular kind of amino acid, and the minimal media that we're plating it on cannot supply it because it's not in the media. So what they did now was they did something really interesting. They took these two mutant strains, and they mixed them together. And when they mix these two together and they plate it onto minimal media that doesn't contain either of the amino acids, no arginine or no tryptophan, they saw to their surprise that colonies actually grew. And this was a big surprise at the time, and no one could really explain why this happened. So another group, let's talk about what our possibilities are, right? So we know at this time, we've talked about two mechanisms of recombination: transformation and transduction. So it's very possible that transformation is occurring in this test tube. The bacteria are dying, their DNA is getting chopped up into tiny pieces, and some of that DNA is getting taken up into other cells, other bacteria, and those bacteria now have the ability to anabolize the amino acid that they were missing. So that's quite possible. It's also quite possible that there's transduction occurring, that there's bacteriophages in this media that are allowing lateral gene transfer to occur. but it's also possible that there's some other possibility, right? We always have to be open to the fact that there's something going on here that we just don't understand yet. I'm sure you can see where I'm going with this, right? So let's look at another experiment. So Davis and his group saw a few years after the Lederberg and Tatum experiment, they wanted to investigate this a little bit further. So they asked that group for their mutant E. coli strains, and they took those bacteria and they put it in this really interesting contraption here. You can see that they have strain A on the left, strain B on the right, but they separated these bacteria by a very fine filter. And this filter had pores in it that were big enough to allow DNA to go back and forth, and also big enough to allow phages to go back and forth. So let's just look at this. I think I have a bigger picture. Yeah. So if we look at this in a little bit more detail, remember bacteria A can't make tryptophan, and bacteria B cannot make arginine. And so if we grow these in the same test tube where the media can flow back and forth quite easily, DNA can go back and forth, bacteriophages can go back and forth, and so on, and then we allow this to, let's say just mix, or I shouldn't say mix because they're not mixing, grow like this for a while, we can plate this onto media that contains, or that is missing, I should say, arginine and tryptophan. And in contrast to the previous, right, in contrast to this experiment where they grew, this one did not grow. So ask yourself what the difference is. What is different in this experiment that wasn't in the original experiment? And of course, they're not touching, right? We aren't allowing those bacteria to touch one another or to come into contact with one another. If it was simply transformation, if transformation or transduction was responsible for this, the DNA and the phages would have been able to get through this filter just fine, and we would have seen growth here. But we don't see growth. So we know that something else has to be occurring. It has to be that third option, something else. And of course, we now know that that something else is this mechanism called conjugation. And they found that there's two things that need to happen in order for conjugation to occur. The bacteria have to be able to touch. They have to, if cell-to-cell contact is inhibited, conjugation doesn't occur. And if DNA replication is inhibited, conjugation cannot occur either. So we know it has something to do with cell-to-cell contact, and it has to do with DNA, which makes sense because we're sharing information. And of course, in the organic world or in the living world, information is DNA. So if you recall, goodness, way back, maybe even like the first week when you guys were all bright and optimistic about microbiology, right? We were talking about the various features of bacteria, and one of these features, or one of these constructs of bacteria, is called a pilus. And some bacteria, they can have lots and lots. You can see all these tiny little pili that are coming out of this bacteria, but most bacteria really only have one sex pilus that comes out of them. So they can have lots and lots of little pili. We sometimes call these fimbriae. But then they only have this one special type of pilus called, sometimes some people pronounce it pilus, some people pronounce it pilus. But they typically only have one. And this pilus acts as like an anchor, and it kind of reaches out and will grab onto another bacteria and bring it closer. So before we get into the mechanism of conjugation and talk about how it works, let's remind ourselves that DNA replication can go in two different directions, or I shouldn't say that. It always goes in the same direction. In other words, the replication is always occurring. The new strand is being synthesized from in a 5' to 3' direction. And we call that, when it's being synthesized continuously, we call this leading strand synthesis, whereas the lagging strand is synthesized in a discontinuous piece because of those, because that DNA polymerase can only work in one direction. So when we have this new strand being developed in the leading strand, it's always synthesizing nucleotides towards that replication fork right here. So something to keep in mind when we talk about conjugation. Oh, let me get straight here. Okay. So it's important to point out that every research group that studies conjugation studies a different model, and gram-positive bacteria conjugate very differently than gram-positive bacteria. And there's even variation within those groups. So all bacterial species have some subtly different, sometimes wildly different forms of conjugation. The one that we're going to look at now, this model, I believe is in E. coli. So we're looking at a gram-negative E. coli conjugation model. And it's definitely the one that's most well known and most commonly studied. And we call this form of conjugation F plus to F minus mating. And we call it F because it has to do with, it's kind of a science. It's actually, I think it's kind of a joke really, but the F stands for fertility. So if you're F positive, you're able to share DNA with someone else. If you're F minus, you're not. So F positive donor cells have a special kind of plasmid within the bacteria that contains genes for the pilus production, replication, and transfer of DNA into another cell. So this particular F plasmid has a certain subset of genes on it, whereas a recipient cell is considered F minus, and they have nothing. They don't have this F plasmid, but they do have a receptor that the sex pilus can grab onto. So let's look at how this happens. So we call this mechanism of transfer, this particular mechanism, we call it the rolling circle method. So remember, let's take a look at this. This is one of the figures from your book. We have two bacteria that are going to share this F plasmid. So the one that's going to donate it or share this information with the other one is going to be called our donor cell. It's F plus. The one that lacks it is F minus. This is our recipient cell. And the bacteria is going to form an F pilus and make that connection with the other bacteria. And then they're going to bring it, basically that sex pilus kind of brings in those two bacteria for very close contact. When this happens, the F plasmid is going to get nicked at a very specific site called the origin of transfer. And we're going to start, the machinery within the bacteria is going to start replicating the plasmid in a 5' to 3' direction. And what this does is it displaces that the outer DNA strand. Remember that DNA is double-stranded, and the strand that's on the outside is going to get displaced by this replication. And what happens here is that 5' end that got displaced is going to make its way into the new bacteria. You can see that here. As it makes its way into that new bacteria, the complementary strand will start to get synthesized in the new bacteria. And then the the F plasmid can recircle itself. And now this F minus cell becomes an F plus cell. So again, super confusing to try to explain through pictures, so let's go ahead and watch a video. Don't want that one. We want, I think it's this one. Let's look. Yeah, it's this one. Okay.
Conjugation is a mechanism of gene transfer that requires direct contact between donor and recipient cells. F plus cells carry the F plasmid, enabling it to make an F pilus and act as a genetic donor. Recipient cells that lack an F plasmid are referred to as F minus. The F plasmid encodes for the F pilus, a protein appendage that attaches the donor to the recipient cell. The first step in plasmid transfer is contact between the donor and the recipient. The F pilus of the donor cell recognizes and binds to specific receptor sites on the cell wall of the recipient cell. The plasmid then becomes mobilized for transfer when a plasmid-encoded endonuclease cleaves one strand of the plasmid at a specific nucleotide sequence called the origin of transfer. A single strand of the F plasmid, beginning at the origin of transfer, enters the F minus cell. A complementary strand to the single-stranded plasmid remaining in the donor is synthesized via the rolling circle mechanism. Once inside the recipient cell, a complementary strand to the single-stranded DNA is synthesized. When F plus and F minus cells are mixed together, eventually all of the cells become F plus.
All right. Make sure we're still doing good on time here. Great. All right. So this is, come on, go full screen here. There we go. All right. So this is considered, again, this is a type of conjugation that is considered the role. It's the mechanism is called rolling circle, and the particular model that we're looking at is called F plus by F minus mating. This happens most commonly in E. coli. But what I want to kind of stress here is that this technically really isn't recombination, because the plasmid isn't incorporating itself into the chromosome of the new bacteria. Right, here's our new F plus bacteria, and it's staying separate. And really what's kind of what's the word I'm looking for? What has been confusing for scientists that study this is that there's really no big advantage to this. So now this bacteria is F plus, and it has this additional plasmid that allows it to share this plasmid with other bacteria. Well, big deal. Like, what does that really mean? It doesn't seem to have any real evolutionary advantage for these bacteria. So it's kind of a confounding question why bacteria actually do this particular type, this F plus by F minus type of recombination. Now, there's another type of recombination that's a little bit more complicated, but it has a much more, we can explain the advantage to this mechanism. So this type of conjugation is called HFR conjugation, and HFR stands for high frequency of recombination. So we still have our F plasmid, but the difference here is that the F plasmid can incorporate itself into the chromosome. This is exactly what I was talking about in the previous slide. So this particular F plasmid has the ability to basically slide into the chromosome and become part of the bacteria's own DNA. And what's interesting here is that it can go into the chromosome, but then it can go right back out again. Notice that look at these arrows. This is actually a two-way street. So here we would consider this cell an F plus cell because we can very clearly see the F plasmid. Here, this cell here we would consider an HFR cell because that plasmid has recombined and become part of the bacteria's chromosome. So what happens in this case? So we can see that we have, so our chromosomal genes are the purple DNA, and we can see two genes of interest, our genetic markers A, B, and C. And we can see that this is an HFR cell because the F plasmid has incorporated itself into the bacterial chromosome. Now, this donor HFR cell is going to connect with a recipient F minus cell with its own chromosome. And what happens here is kind of interesting. So we still have our origin of transfer. That origin of transfer is going to get nicked, and the bacteria is going to, or the DNA rather, is going to start making its way into the other bacteria. But notice that now it's not going to get all the way through, because now it's actually trying to get the entire bacterial chromosome through that sex pilus. And there's simply not enough time for that to happen. In order for that entire bacteria's chromosome to be replicated and put into, like in a single strand like this, it would take, I think they estimate, around 100 minutes, and it never lasts that long. So what happens here is when the piece of DNA is starting to get shared, only some of it gets through. And what's interesting is that the genes that are closest to that origin of if here's our origin of transfer, the genes are going to go in, are going to get transferred in a certain order: A first, and then B, and then C. So in other words, the closer a gene is to the origin of transfer, the more likely it's going to get, get to the other bacteria before the sex pilus breaks off. So here we can see that after the transfer ends, only a certain portion of this bacteria's chromosome got into the new bacteria, and the sex pilus broke off, and they're no longer successfully conjugating. And if you can see, what happened here is that the entire F plasmid didn't make it across. So this cell is not an F minus, or is not an HFR or an F plus cell, but what happened instead is that this bacteria picked up a couple of new chromosomes. It picked up, it picked up a couple new genes. It picked up gene A and gene B. So this is considered HFR conjugation. So let's watch, again, very confusing to try to explain with pictures, but let's watch a video.
Sometimes the F plasmid becomes integrated into the host cell genome. The host cell is now referred to as HFR, which stands for high frequency of recombination. The HFR is able to transfer some of the host genes to the recipient. The sex pilus contacts the recipient F- cell and pulls the cells together. The donor chromosome is transferred as single-stranded DNA starting at the origin of transfer. Gene 1, which is closest to the origin, is transferred first. Segments of the integrated plasmid are at the beginning and the end of the DNA being transferred. It is theoretically possible for the complete genome and the F plasmid to be transferred to the recipient cell. However, this does not happen because the donor and recipient cells will separate prior to the complete transfer of the donor chromosome. The transferred DNA becomes double-stranded. The donor DNA is integrated into the recipient cell's DNA by homologous recombination. The recipient now carries transferred genes, but remains F-, whereas the donor cell remains HFR.
All right, hopefully that was easier for you to understand than trying to explain it picture by picture. So now, if you think about what is happening here, we now know the relative locations of these genes within a chromosome. If the origin of transfer is here and it's replicating its DNA in this direction, we know that A comes first and then B and then C. And what this allowed scientists to do is actually map the genes of bacteria. So let's talk about this experiment. It's called, I think it's kind of a funny name, it's called the interrupted mating experiment. And it's a fun logic puzzle that allows us ultimately to figure out the relative positions of genes on a bacteria's chromosome. So imagine if we have, this is easier to remember if we were back at our benches in the micro lab, but let's imagine that we had 4 people that were sitting at one of our bench tops, and you had these 3 pieces of information. Susie is to the left of Angel, Angel is diagonal to Edna, and Edna is to the right of Martin. Can you figure out where the students are sitting based on that information? Well, you can actually. You can come up with 2 possibilities. We have Susie is to the left of Angel, Angel's diagonal to Edna, and Edna is to the right of Martin. So we know their relative positions. However, it could be like this. So both of these situations are true, and that's why we are very careful to say the relative locations. We know where these 4 students are sitting in relation to one another, but it could be this one, it could be these positions, or these positions. We're not totally sure based on this information. So what does this have to do with bacteria? Well, let's look. Okay, got lots of information, a lot of writing on this page. So let's just take it piece by piece. We want to know the relative location of 3 different genes. The genes that can make the amino acid threonine, the genes that can make the amino acid arginine, and the genes that can make the amino acid methionine. We have 2 types of bacteria. We have our donor bacteria, our HFR donor, and we have our recipient bacteria, our F- bacteria. Now, our donor bacteria is going to be prototrophic. If you recall, prototrophic means it can make its own nutrients. It can make its own, in this case, amino acids. It can make threonine, arginine, and methionine. However, it's sensitive to the antibiotic. In this case, we're using the antibiotic streptomycin. So if we were to write down the genotype for our HFR donor, we would say it's threonine positive, arginine positive, methionine positive, and streptomycin sensitive. Now, on the other hand, we have our F- recipient bacteria, and this bacteria is going to be auxotrophic for those same genes. It can't make any of these three amino acids. However, it's resistant to the bacteria streptomycin. So it is able to grow on media that contains streptomycin. We allow these we allow these bacteria to mix. So we put them in the same flask. We mix it so that they are allowed to come in contact with one another, but we're not going to allow them to stay in contact for very long because we want to actually time it to see which genes will be transferred first. We know that the genes that are transferred first will be the ones that are most closely positioned to the origin of transfer. So let's kind of visualize this a little bit. We have our HFR donor. Remember, prototrophic, sensitive to streptomycin. Our F- recipient is auxotrophic, but resistant to streptomycin. And we're going to mix these two strains of bacteria together in a flask. And we're going to allow conjugation to occur. And we're going to remove samples at specific times and plate them onto 3 different types of media. Let's look at our media that we're going to plate onto. We have media, all the media contains streptomycin, antibiotic streptomycin, but each of these media are different in that each of them are missing one of those amino acids. This media is missing threonine, this media is missing arginine, and this media is missing methionine. So what happens here? Let's just quick think about before conjugation occurs, could we see, should our HFR donor and our F- recipient bacteria, can they grow on the following types of media? Remember, CM stands for complete media, and streptomycin is our antibiotic of choice. So pause the video and see if you can complete this table. So let's look at our HFR donor first. We know, of course, that our HFR donor is, even though it's prototrophic, it can't grow in the presence of streptomycin. So of course it can grow on complete media. It could probably grow on minimal media too, but it's going to grow just fine on our complete media. But anything that contains streptomycin can't grow on. So we know that if we plate things onto streptomycin, that our HFR donor cells cannot grow on that media. On the other hand, our F- recipient can, it's auxotrophic, but it's resistant to streptomycin. So it can grow on complete media because remember, complete media supplies all the nutrients, and it will grow just fine on complete media that contains streptomycin because remember, it's resistant. However, it cannot grow on any media that is missing any of these amino acids. So here we have minimal media that's missing threonine, minimal media that's missing arginine, and minimal media that's missing methionine, for example. Can't grow on those because the bacteria can't make it and the media is not supplying it. So if we take the conjugation, remember let's go back here. Remember we took these 2 bacterial strains, we mixed them, we allowed conjugation to occur. And if we see colonies that are able to grow on minimal media that does not have threonine in it, we know that this bacteria must have a certain quality to it. Likewise, this bacteria must have gained something, and then this one on the right must have gained something too. So ask yourself, what does this bacteria have to have if it can grow on this media? Well, hopefully you recognize that the bacteria that are growing on these plates has to be that F- recipient. It has to be this bacteria, the F-, because it's the only one that's resistant to streptomycin. So we know that these bacteria can't be our HFR donor bacteria. It has to be the F- bacteria that has gained the ability to, in this case, synthesize threonine because it can grow on media that's missing threonine. Bacteria that are plated on this media must have somehow gained the ability to synthesize its own arginine. Otherwise, it wouldn't grow. And finally, bacteria that can grow on this media must have gained the ability to make methionine. So by separating out these 3 amino acids and selectively adding them back to the media, we can see which bacteria gained which gene. So we can actually time this, right? So we can pull, right, we're going to take bacteria out of this flask at time 0, at time 3, at different time periods after conjugation. And as you can imagine, if we were to mix these bacteria and immediately pull the bacteria out, we shouldn't see any growth in any of those plates because conjugation can't occur in 0 minutes. It takes longer than that for conjugation to occur. But interestingly, as soon as 3 minutes after adding and mixing those two bacteria together, we're going to start to see gene transfer from that HFR donor bacteria to our F- bacteria. So we can see at 3 minutes, the bacteria will start to grow on the threonine minimal media. At 6 minutes, we start to see the methionine strain start to grow. And then finally at 9 minutes, we start to see the arginine strain start to grow, bacteria start to grow. And then, of course, at 12 minutes, all of them are growing quite nicely. So what this means is that we can actually time the ability, the time it takes for conjugation and replication to occur. And it's sensitive enough that we can separate genes that are 3 or more minutes apart on our bacteria's chromosome, which is I think that's kind of neat. So let's look at this in reality. So here's our chromosome. Here's a representation. Remember that bacterial DNA is circular, it's double-stranded and it's circular. And here's our origin of transfer. And so we know that as our origin of transfer gets nicked and it starts to conjugate into our F- bacteria, which would be over here, that threonine gene had to be closest to our origin of transfer because that's the one that got through first. This got through in 3 minutes. This one got through in 6 minutes. And then this one was the last to get through in 9 minutes. So theoretically, we could take, this could keep going and we could see the order of all of the genes on this particular plasmid, but we know that it will break off, conjugation will get interrupted before that. So what ends up happening is we have multiple origin of transfers. We could do one here at the X spot, we could do one at the I and at the O, just to see, sorry, I got an itch. Okay. Just to see where, just have multiple sites of origin so that we can overlap the order of these genes so we have more confidence in our findings. So let's look at some real data. This is actually data from one of those original experiments that we're trying to map the chromosome of, I think this is E. coli. Yeah, it's E. coli. So we can see that time after conjugation, right? We have 5 minutes, 10 minutes, 15, 20, and so on, that the first gene to go through was B, the second gene to go through was A, and then E, and then at some period of time after C, and then D. So we know that this particular experiment can be represented by this line here, B, and then A, and then E, and then C, and so on. So if we repeat this several more times, we're going to get different data because we're using different origin of transfers. So in the next one, we're going to use a different origin of transfer. And in this case, the F gene came through first, and then B, and then G, and then E, and so on. And so you can see we repeated this 2 more times. And if we overlap our findings, we can see how this gene or how this chromosome is actually constructed and the relative order of these genes. So you can see that the F in this particular kind of sequence, the F plasmid comes first. So we're going to set F as our kind of our noon. Like if we're looking at a clock, F is right here at the top. And then the next one to come through would be B. And then it looks like A. And then going down G, and then E, and then H, and then C, and so on. And so we can map the relative locations of these genes. And so before we had the ability to just throw some bacteria in a sequencer and find all the nucleotides that made up this chromosome, we had this really inventive way and this very creative way of mapping the relative locations of genes on a bacterial chromosome. So let's ask some questions just to make sure, because this is a very complicated, if you don't get this right away, don't feel bad. It's going to take some time. And I'm happy to kind of go over these questions with you in a Zoom meeting or something if you want to work through these together. But let's talk about some concept questions so that you can kind of get a sense of how you're losing my train of thought right now, how you are getting the concepts behind the interrupted mating experiment. So what is the purpose of this experiment? Again, pause the video, ask yourself which is the answer. Remember, well, I would argue that D is also true. Or, I'm sorry, D and E are both true. It's definitely, we can use it to demonstrate that bacteria are able to share genes with one another, and thus recombination can occur. It's also always fun to mess with bacteria, but it really, the value, the value of the interrupted mating experiment is that it's allowing us to map genes on chromosomes. We can see what the relative locations of these genes are. So remember, we always have two different bacterial strains that we're going to use in an interrupted mating experiment. What would be an appropriate genotype for a recipient cell? Well, remember, our recipient cell has to be resistant to a certain antibiotic of choice, so that when we plate it onto antibiotic-containing media, that the donor strain can't survive. So we never want the donor strain to grow, because remember, the donor strain is prototrophic. It'll kind of cloud our interpretation of the results if we allow our donor strain to grow on our plates. So we want to put an antibiotic on our plate that only our F- strain is sensitive to. So we can rule out anything, let's see, we can rule out C and D because both of these are sensitive to the antibiotic that we're going to use here, which is ampicillin. So we know that it can be resistant to ampicillin, so it's either going to be A or B. And A is prototrophic. It can make its own amino acids, in this case, methionine, histidine, and tryptophan. And that's not going to work because if it can make everything on its own, it doesn't need anything to grow. In other words, this bacteria is prototrophic, which wouldn't work. So in this case, the answer is B. It's auxotrophic for these three amino acids. All right. What genotype is that of a recombinant cell in an interrupted mating experiment? So I really hope these are going to be questions on the test, right? You're going to have this kind of experiment on the test, some variation. So if you're not pausing it and you're just kind of listening to me talk and you think that you can kind of get it, I would strongly discourage you from that idea. Students really struggle with this, so pause the video, try to do the work yourself, and see if you get it right. So if it's recombinant, we know that it has to be resistant to ampicillin, right? If it's sensitive, it's not going to live. And in this particular case, it had to have gained something. So the answer here would be A. It gained the ability to make histidine. So it could grow on minimal media that is missing histidine, for example. So we know that recombination has occurred because it gained the ability to make something, in this case, the amino acid histidine. Okay, so here are the genotypes, just for your own reference, so you don't have to kind of go back and forth. Here are the genotypes from that previous question. What kind of media would the one that's highlighting in green, which media would grow only recombinant cells? This is a very tricky question, so see if you can get this one right. What is the answer? Okay, so there's actually two medias that would... Actually, hold on, let me think about this for a second. Okay, so let's start with C. so we have minimal media that's supplemented with methionine plus ampicillin. So because it contains ampicillin, we know that it would only grow the F- cells, but these particular F- cells wouldn't be able to grow in this media because the bacteria can't make methionine, it's methionine negative, and this particular media does not contain methionine. So this one would grow nothing. Let me see, let me grab my, stand by for just a second while I grab my pen. Where's my pen? Okay, I don't know if this is going to work or not. So we have our, oh yeah, there you go, our HFR and our F- cell. So these are our two strains, and in theory, we did a recombination event that mixed these two strains. So in C, neither of these plates are going to grow. In other words, our HFR can't grow because it contains ampicillin, and our F- cells can't grow because it can't make its own methionine. So now let's look at minimal methionine without ampicillin. So we have our two plates, and if it doesn't contain ampicillin, our HFR cells are going to grow on there. So we're going to see our HFR cells grow on the ampicillin just fine. However, our F- cells will not grow. This media will stay blank because our F- cells can't make methionine and the media itself is not supplying it. Now let's look at B, minimal histidine without ampicillin. So if we have the media that contains it doesn't contain histidine, but it also doesn't contain ampicillin, our HFR cells are going to grow because it doesn't contain ampicillin. Our F- cells are also going to grow because it has it can make histidine. So as you can imagine, as you have now figured out, of course, A is our correct answer because what happens here? If we have our two plates, our HFR is not going to be able to grow on here because this media contains ampicillin, whereas our F- strain will be able to grow because it's resistant to ampicillin and it can make its own histidine. So in this case, answer A is correct. Oh, wow. Guess what? I have a surprise for you. We're done. So I decided to cut CRISPR out of this lecture and of course out of the exam because the CRISPR technology was what we did our group work on, and because we can't do group work for exam 3, I decided to leave it out of the lecture. So that does it for this, not only this lecture, but unit 3 in general. I want to show you really quick. I got rid of, I'm sure lots of you, no, I don't want to do that. I'm sure lots of you already printed this out, so I just want to really quick point out that I did cross out those learning objectives that relate to the CRISPR technology. Unfortunately, I think it's just too difficult and too complicated of a concept to teach online. So maybe when I get better at teaching online, we can do it. But for now, less material on the test, which is always a good thing. So we only have 2 lectures for the Unit 3 exam. We have lecture number 8 and lecture number 9, and you now have all the information that you need for the test. So that does it for the lecture portion, and I will see you next week.